So now before we begin with specific requirements of different types of cultures, let's see in order to redifferentiate and de-differentiate what happens in the plant cells and what is needed. So whenever you bring an explant, now explant is a tissue or an organ which you have brought. So it is a mixture of many different kinds of cells, isn't it? So if you want that cell to come back to, now it is a differentiated cell performing a specific function. Now if you want to bring back the cell into some other form of tissue or organ, the first that cell has to be reprogrammed. Although all the genetic information in the plant cells, as we say, they are totipotent in nature, which means that they have the capacity to redifferentiate into some other form. So which means in turn all the genetic information available for carrying out those metabolic activities is available. It is just that at a particular time there is only specific sets of genes which are expressed. So under in vitro conditions, when you bring an explant, first thing, the cells within that explants have to be reprogrammed. Now that reprogramming should be involving two things. One is cytodifferentiation and the other is organogenesis, in order to do regeneration from that explant into some other form. So what are the kind of cells in an organ or a tissue which you bring out from the nature? These are non-dividing, differentiating, quiescent cells. Now they are matured, so now they are performing a specific function. So they are non-dividing generally, these cells of the explant. So when grown on a nutrient medium, they first undergo changes to achieve the meristematic state. So these programmed cells or matured cells have to be reprogrammed to come back to their original meristematic state. Now the phenomena of mature cells reverting to this meristematic state and forming an undifferentiated callus tissue is what is called as de-differentiation. So if you know, we were saying that callus is a de-differentiated tissue. So the process of reverting back a matured cell into a meristematic cell is called as de-differentiation. That is what forms that callus blob. Now since the multicellular explant comprises cells of diverse type, the callus is therefore heterogeneous in nature. Now heterogeneity arises due to two factors. One is inherent variation in the parent plant material, because there are so many different kinds of cells. The second is cytological and genetical differences. Now all these cells are performing different functions, which means that some amount of genetic information is expressed and some genetic information is not expressed in these different types of cells. Now the ability of the component cells of the callus to differentiate into the whole plant is what we know as redifferentiation, or the totipotency is the capability so as to de-differentiate and redifferentiate. It is not necessary that the explant has to go to a callus stage before regeneration. So now the cells in a callus, we know they are parenchymatous in nature, which means generally all these cells are parenchyma cells. Now the differentiation of these cells to a variety of other forms of cells needs the cells to be redifferentiated. Now this redifferentiation stage 1 is called cytodifferentiation. So in vivo or in vitro, in plant cytodifferentiation, main emphasis is forming these vascular tissue. So which is, for example, as I said, xylogenesis, forming of phloem and xylem elements through their connections. Now various factors, for example various chemical and physical factors are responsible for this kind of differentiation. For example, you can take auxins, cytokinins, carbon source which is sucrose or gibberellins. All genes, as I said earlier, all genes which are responsible for this process, now the cells being totipotent, all genes are present, but due to the environmental conditions or the stimuli present in vitro, some of these cells, the genes will get expressed for the reprogramming to happen and the cells to re-differentiate into some other forms of cells and thereby forming another type of tissue. Now, for development of an adult organism from single isolated cells from these differentiated tissues, which are generally non-dividing and quiescent cells, the differentiated cell first undergoes de-differentiation and re-differentiation. Now, when I say cytodifferentiation, one is that formation of vascular bundles. Now, then and it has to, in order to regenerate into a whole plant, what is needed? There has to be a shoot bud formation or primordium, which is called as. Now, once this is done, shoot bud primordium would form directly, then it is direct regeneration happening, or somatic embryos are formed through somatic cells. If somatic embryos are formed, then subsequent development of bipolar structures, which is shoot primordium and root primordium, will form. So, organic differentiation can be of two forms: direct formation of shoot bud or root bud, or formation of somatic embryos, thereby then forming bipolar structures, one leading to shoots and the other leading to root. Now, regeneration, how is it useful in comparison to whole plant reproduction through which progenies are produced? There are three ways in which it is useful. It is more efficient process. You can do clonal propagation of the species, which means less time taking, and what else? The potential for the production of much higher number of plants. So, the number of plants, the rate of production of the progenies is also enhanced. So, this is how under in vitro conditions, if plant biotechnology tools are used for regeneration, then these are the advantages in comparison to natural plant production. Now, organized development is successfully achieved through proper selection. So, what things have to be kept in mind under in vitro conditions for regeneration to happen? You need to have a good starting material, which means the inoculum. Then proper choice of the medium, the media composition matters. Then a balanced composition of growth hormones, which means what kind of the effect of auxins, cytokinins, gibberellin, as I said. So, these have to be optimized, and moreover it is controlled physical environment. Now, what happens in organogenesis? The cells are forced to undergo changes which can lead to the production of unipolar structures, as I said, generally shoot. So, once you will see that when shoot multiplication is done for regeneration, it is shoot bud primordium which comes out, you get multiple shoots, and then later you put it into a root inducing medium such that now these shoots will have roots and before they are transferred to the fields. So, in organogenesis, shoot bud primordium is the objective when you are trying to regenerate into new plantlets. Now, root and shoot differentiation is a function of quantitative interaction between auxins and cytokinins. Whether auxins, so last class also, as I said, that depending on what species are you working, whether dicot, monocot, herbs or tree species, the effect of the same auxin may vary. Now, in general, what has been found, that higher ratio of auxin to cytokinin would lead to a production of root, and lesser ratio will induce shoot formation. Now, usually high cytokinin concentration favors shoot formation, as I said, and 2,4-D is generally used for cell proliferation, but it suppresses cellular and organ. So, these things you can note down, although this is species specific, but in general how it helps, that when you are working with a species for which no information is available, this can be the starting point. Now, 2,4-D promotes cell proliferation and suppresses cellular and organ differentiation in dicot plants. I hope you know what are monocots and dicots. Monocotyledonous, dicotyledonous, cotyledons, that much you people know. Now, abscisic acid and gibberellins. Now, what is abscisic acid and gibberellins? These are plant growth regulators, plant hormones. Now, they are also sometimes used to be promoting organogenesis in the medium. Now, sometimes nitrogen, complex nitrogen sources like casein hydrolysate and amino acids are also known to affect shoot bud formations. Now, light quality, intensity, period, which means photo period, then temperature can also affect organogenesis in in vitro cultures. So how does the organ development begin? Differentiation begins with the changes in the individual cells in response to the external stimuli. So generally what happens, that if suppose there is a cell which is competent enough for de-differentiation to happen, so there are competent cells which are ready for differentiation, de-differentiation, which means meristematic, which have the ability to quickly go through meristematic state. So it begins from there. Around that cells there develops many meristematic centers. So from these meristematic centers the development of organogenesis takes place. So there is a cell which is determined to undergo differentiation and de-differentiation. Around that cells, after multiplications, there are some more meristematic centers which get formed, and these meristematic centers together will lead to formation of your shoot bud or root bud primordium. Now the morphogenetic signals set up the conditions which allow competent cells. So these are called as competent cells. So not all cells, and sometimes it is very species specific. Sometimes you may try out many hormonal combinations, but you will see that you are not getting successful organogenesis happening. So which means that the explant, it depends on the nature of the explant, the age of the explant, the season in which you have collected the explant. It may not end up, the cells in that explant may not be competent for coming back to a meristematic state and then re-differentiating. It also depends on the environmental conditions which you have given. So there are multiple factors which are responsible for successful regeneration to happen, and even the genotype and the type of explant which you have used will be playing a crucial role in this. So why did I say? Because there has to be cells which should be competent enough to undergo this reprogramming. So morphogenetic signals set up the conditions which allow competent cells to undergo internally this controlled program of differentiation to happen. Let's talk about somatic embryogenesis. The process of embryo development from somatic cells is called somatic embryogenesis. Now when we say somatic cells are non-germ cells, the cells which do not participate in the sexual reproduction of the plants. Now somatic embryos can be formed from callus, cell cultures, protoplast, or any kind of organized structures from the plant. Now plant regenerations via somatic embryogenesis can be divided into two phases. One is inductions of cell with embryogenic competence. Now this happens when you have very high concentrations of auxins in the medium generally, and the development of these cells subsequently into embryos once vascularization, which means cytodifferentiation and organogenesis both have taken place. Now once organogenesis also has taken place, which means coming up of the shoot and the root primordium in these. So for subsequent growth you need a combination of cytokinins and auxins. Now 2,4-D is the most commonly used auxin in somatic embryogenesis. Sometimes thiodiazuron or zeatin, these are other kinds of cytokinins which may also help in subsequent growth of development of somatic embryogenesis, embryos, because even in the development of somatic embryos there are different stages involved. We will take up that later once we come to somatic embryos. They are first globular structures, then torpedo, heart shaped structures, though this is a gradual development. So for this gradual development also you need variation in the nutrient. So abscisic acid is used in combination with cytokinins for maturation of these somatic embryos. Now sometimes in some species even activated charcoals can impact the induction of somatic embryos. So it is the stimuli which can induce either the determined cells for this maturation to happen, or some of the cells will be induced to undergo embryogenic changes. So a high nitrate to low ammonium nitrogen also has been found to favor somatic embryogenesis. Reduced nitrogen is required during embryo development. So induction is one thing and subsequent development is another. So it is not necessary what has worked for inducing will work for subsequent development. If you see that once callus has induced or somatic embryo has induced, you may find that keeping in the same medium does not lead to further growth, even if it has led to successful induction. So which means that it needs revision of the media. Okay, somatic embryogenesis of single or multi-celled origin is known to occur. Now they can occur even from single cells or multiple cells together. Somatic embryos of a single cell origin, they are produced from synchronous cultures. Synchronous cultures means all the cells have the similar metabolic activity. Now somatic embryogenesis can be induced directly from the explant or can be indirectly from the callus phase. So in case when the callus phase is involved, once you bring the explant, then it develops into callus, then you do a variation and it develops into somatic embryos, that is called as indirect somatic embryogenesis. And when directly an explant, which means a leaf giving rise to directly somatic embryos, then it is called somatic embryogenesis, direct. It is presumed that the cells of the explants have the competence and determined to produce somatic embryos. Now there are two kinds of cells. What is the hypothesis? The explant tissue contains cells that are already determined to embryogenic development. So these are called as pre-embryogenic determined cells. These group of cells are already determined that if given the chance they will form somatic embryos. Others are called as redetermination through a period of culture, which means induced embryogenic determined cells, which means they can be brought into somatic embryogenic stage but after reprogramming, which means keeping them under the right stimuli so as to reprogram them into, induce them into somatic embryogenesis, which means that it will take much longer time. Now embryogenic cells, they produce embryos more readily than the differentiated explants or induced embryogenic competent cells. So it is presumed that highly differentiated cells, more epigenetic changes. So when we change the environmental parameters or we give stimuli, then what is happening? The cells are undergoing these genetic changes in the sense the genetic machinery which was not expressed then starts getting expressed, which means which was cryptic is now expressed, thereby leading to some DNA replication and translation, thereby leading to biochemical changes, histological changes and cytological changes, which then you can see the morphogenetic events like somatic embryos. Now it is presumed that highly differentiated cells require more epigenetic changes. Now what do you understand by epigenetics? Right, which can bring about a change in the gene expression. Now in some species it is not possible to induce such epigenetic changes, and it depends on as I said in the nature of the explant, and also depends on the conditions which you give in vitro. If those conditions are not adequate enough to bring them into induced embryogenic stage or to bring about such epigenetic changes, then you won't get successful results. Now what are the factors which can affect regeneration? It is the type of the explant, sometimes the size of the explant, season of collection, age of the explant. So younger plants and younger explants, they can easily get into and can be manipulated and they are more conducive for epigenetic changes. Reason? Because they are still dividing cells. The majority of the cells will be at a meristematic stage because they are younger. They have still not completely differentiated and become organized to carry out a matured function. Now they can be easily manipulated. The explants can give rise to organs and embryos directly or indirectly via callus. So as I said, what happens that there is a single cell, there may be a single cell which is determined to undergo cytodifferentiation and organogenesis. As it divides, there are multiple meristematic centers around that region. These meristematic centers undergo reprogramming and lead to your shoot or root primordium formation. So what kind of nutrient media helps in regeneration? Inorganic micro and macronutrients, major difference, as we said yesterday, we were talking that although defined media can be used, but there is a difference in these. So, if it is high ammonium to nitrate, so generally what induces somatic embryogenesis? When it is a very high nitrate to ammonium ratio, or suddenly you give very high concentrations of sucrose. So, or if you can increase the concentration of auxin. So, although dependent on species, but in general it may be noted, if no information is available, these things can be tried, and rest of the environmental factors anyways can play a role in generating these organized structures. Okay, that's it. I will stop here today.